This is a freshwater sample, and this is a trumpet snail under the microscope from that freshwater sample. In this video, I'm going to show you how I collected these samples and what I found when I put them under the microscope. I attached this plastic shovel to a selfie stick, and I used it to collect a wall of macroalgae. I also used this baster to collect some sediment and place it in a jar. Sediment from a freshwater body contains a lot of microorganisms, including ciliates, nematodes, and diatoms. I also used that same shovel attached to a selfie stick to collect some macroalgae and plant matter. Then I placed them in a jar. This sample is the one that was shown in the beginning of this video. Macroalgae from a freshwater body contains a wide variety of organisms, including snails, flatworms, and ostracods. I ended up collecting 5 samples in total. I began observing the samples by placing some of the macroalgae under the Microscopy Series 305 stereo microscope. This is one of the trumpet snails that I collected. Trumpet snails are a common type of freshwater snails. They feed on algae and decaying organic matter. Trumpet snails get their name from the shape of their shell. Apparently, they are common in the freshwater bodies that I collected the samples from. Back to the macroalgae. When observing it using the Swift 380T biological microscope under high magnification, we can easily see the chloroplasts, which are the green spheres seen here. They are used to perform photosynthesis, meaning that these chloroplasts are used by the macroalgae to absorb carbon dioxide and water to produce sugars, releasing oxygen in the process. While this might look like more macroalgae, it is actually a filamentous colony of Oscillatoria, which is a genus of cyanobacteria. Like macroalgae, cyanobacteria can also perform photosynthesis. It can be challenging to see when filmed through a phone camera, but this tiny dot is actually an ostracod. Let's take a look at it under the microscope. Under the microscope, it is hard to follow the ostracod, which is because it is very fast and active. Ostracods are sometimes called seed shrimp. They earned this name due to the shape of their shell. A lot of ostracods are benthic, meaning they can be found crawling on the sediments in the bottom of the pond. Other ostracods can be found crawling on macroalgae. And while ostracods seem to be common in this freshwater body, I was never able to observe them under the Swift 380T microscope before. While I was able to observe freshwater ostracods using a digital microscope, and marine ostracods using this microscope, but never freshwater ostracods using this microscope, until now. Another organism found in the macroalgae is a flatworm. Flatworms have a flattened shape in order to allow oxygen to flow into their bodies. They take in oxygen through diffusion, because they don't have a respiratory or a circulatory system. I also found quite a lot of these, which are the larvae of some kind of flying insect, most likely a midgefly. We can also observe much smaller organisms, including this rotifer. Rotifers are microanimals, meaning they are multicellular organisms, but they are smaller than the other animals shown in this video so far. This is another rotifer. It looks very different because, unlike the previous rotifer, which belongs to the class Deloidea, this one belongs to the class Monogononta. This worm is another microanimal. It is known as a nematode worm. I find nematodes in almost every sample that I collect, whether it's moss, freshwater, or soil. This is a colony of paratric ciliates. Their bell-shaped bodies are attached to stalks, and these stalks are attached to surfaces. They contract rapidly when disturbed. Paratric ciliates have a ring of cilia around their bodies. This ring is used to draw in food particles. Back to snails. This trumpet snail is eating some macroalgae. This other video also shows a trumpet snail eating macroalgae, but at a much higher magnification, meaning we can see the snail eating up close. But also, we can see that the snail eats using a tongue-like structure known as a radula. This is another snail. While I am not certain of its identification, it may be a ram's horn snail. I think so because of the shape of its shell. Now, let's observe one slide of material from each sample and see what we can find. In the first sample, we can see a lot of macroalgae. And we can also see a rotifer attached to this macroalgae. This rotifer is filtering the water for food particles. Another noteworthy thing is that some strands of macroalgae are covered in cyanobacteria, which are photosynthetic, just like the macroalgae. Let's move on to the next sample. This sample is the one with balls of macroalgae. Aside from the large amount of macroalgae, I was able to find these rotifers. One of them is a deltoid rotifer, and the other one is a monogonont rotifer. The smaller rounder one is the monogonont rotifer. I also saw this weirdly shaped ciliate. It was swimming around and hiding in the macroalgae. On to the next sample. This one consists of mostly stones and algae, so I used a pipette to collect some of the algae that settled in the bottom of the jar. Then, I placed it on a slide to observe it under the microscope. Here is what I saw. Firstly, this ciliate. It likely belongs to the genus Euplotes. And secondly, this paratric ciliate. Unlike the other ones, this one is on its own. It is not a part of a colony. Thirdly, this rounded diatom. Diatoms produce a significant portion of the Earth's oxygen. Next, let's take a look at some of the sediment under the microscope. I only put a small amount of sediment, because it separates between the slide and the cover slip, allowing microorganisms to swim out of focus. So, putting a small amount helps reduce this space and keep microorganisms in focus. In the sediment, I found diatoms. This one is much longer and pointier than the one shown earlier, leading me to conclude that it's a different species. This is what it looks like when illuminated using dark field illumination. Let's take one slide from the last sample. This one consists primarily of microalgae. I collected it from the sides of the freshwater body. I placed a few drops of it on a slide using a pipette. Then, I covered it with a cover slip. Under the microscope, we can see that the primary inhabitants of this sample are rotifers and cyanobacteria. I also saw this euglenoid, probably Paranema. And that's the end of the video. This video covers all what I was able to find in these freshwater samples. If you are interested to know what I was able to find in most samples, click the video which is shown on the screen.